Okay, we start the recording. It is July 31st, Bio 105, summer 2025. And we are here at the end of week 4. Okay, so let's look at the schedule. We finished our week 4 PowerPoint yesterday. So we ran through mitosis and meiosis. That's cell cycle. Okay, wrapped up. It's one of the biggest things you gotta know when you hit the upper division, at least like certain science, upper division science courses. You really need to know that stuff. We're going to now move into the week 5 PowerPoint today, where we'll be talking genetics. Okay, and we'll wrap it up at the end of, at the end of next week with a little bit of evolution, natural selection stuff. Okay. So, next week we have our lab final and our lecture final exam. And I have finally finalized the finals, okay? So I have a better idea for you of what they are going to look like. The lecture final, okay, is 40 multiple choice questions instead of 33, 2.5 points each, right? So each question is now worth a little less of your final 100 points, okay? You get a full hour and 50 minutes to complete that next Thursday, a week from today, all right? It's comprehensive. It's cumulative is the word I'm looking for. I always say comprehensive when I mean cumulative. And the study guide is posted, okay, in the week 5 module. Yes?
Did you make any changes since like a week ago or something on the study guide?
No. No, it's all the same topics. All the same topics are covered. A couple of questions have been changed because I was like, this question was not good or too vague, Yes, please. Great question. I'm going to repeat it for the recording because some people will be watching that. Should you use a study guide from just week 5 or both study guides? The study guide from week 5 has everything on it. Now, if you've already completed your study guide, if you filled out your study guide from the midterm, you'll notice a bunch of the topics are the same. So you can totally just port over information into the final study guide, assuming that that you felt those topics were fully explained in your midterm. There are some questions that I have just lifted directly from the midterm and copied and pasted into the final exam. All right? Some of them, I just changed a couple of numbers, but they're essentially the same question. Okay? Looks like I'm just going to give you all the details here. Questions 1 through, what would I say here? 1 through 13 are from pre-midterm, right? That means questions 14 through 40 are post-midterm. So, try to stick to that 70-30 balance, 70% new material. Kind of makes sense? Awesome. Everything that you need to know is on the study guide, or all the topics that are going to be covered. The lab final, here it is. It's significantly longer, right? It's more pages. It is 25 questions. However, many of these questions are like question 7. You'll probably, if you know the answer to question 7, you're going to be done with it in 30 seconds, and it takes up a whole sheet of paper. Questions 12 through 17 are multiple choice or true-false questions. All right? That's pretty nice. Yeah. There's a Hardy-Weinberg on there. There's some dilutions and solution making. Next week, I'll do the same thing I did for the previous, for the lab midterm, where I sort of go through all the topics that you need to understand, I'll make some extra time for that on Tuesday, because Tuesday's lab period is set aside, as you'll see, for reviewing for the lab final. All right? Solid. We've got a whole day for it, and for looking at bacteria. Questions?
we have lab on Wednesday? Or no, Wednesday would be our lab quiz.
Wednesday's your lab final, so there's no lab on Wednesday. Tuesday's lab final review and observing transformation results. So it can run as long as the students sort of let it go. Because I'll run through the topics, and if everybody's like, okay, cool, we're done, then we'll just end. It'll be short, you know? So the only lab that we actually have next week is on Monday, and it's gel electrophoresis. And I kind of, I thought about how important gel electrophoresis is, and I'm gonna have each bench run a gel, even if you just have a few samples, and we'll look at your ALU results then. I think and we'll do restriction enzyme digestion, which is all important stuff. We'll set aside a whole day for it. The gel takes an hour to run. We'll probably be able to go set up the gel, run the gel, interpret the results, and be wrapped up for Monday. I'm giving you way more information than you need, I think. Did I answer your question? Cool, probably like 5 minutes ago, right? Okay, yes, please, question.
For the quizzes next week, on there it says due August 3rd.
Oh, I adjusted that. Yeah, thanks. It's one of those things where honestly, the course schedule, what I do is I just, what's the word I'm looking for? I copy and paste it in, change all the dates, and I missed this. Yes, please.
But I think it's chapters 17 and 16 is a quiz, but you don't have a due date for that. Are we still doing that quiz? Or
Quiz chapter 17 and 16, no, we're not doing those. This happens with copied Canvas pages, they'll end up hidden in my quiz folder, and they just still exist, 16 and 17, and somebody's completed it already. So because somebody completed it, the only way I can do this is deleting the quiz. And that person did work for nothing. If you're in this room or if you're watching this recording, I'm very, very sorry. Good job being so on top of the quizzes and stuff. Yeah, that has happened in the past, and I always have to just delete that thing. It stinks. Okay, cool. Any additional questions? Thank you so much for catching that, by the way.
you're welcome. I was asking about it yesterday, but there was no time, so I'll just ask right now.
It worked. Everything worked out. And yeah, the student who did the chapter 16 and 17 quiz, I think it was like maybe it's probably this one person. If you come talk to me, I'll trust you. Maybe I can find a way to like give you an extra credit point somewhere or something. Question?
Do we need to have like our own chi-square table for the exam? Or?
For the lab final, it will be provided to you.
Okay.
Yep, it's provided to you on the exam. It's very small,
But it's what we need.
but it's what you need. It just has degrees of freedom 1 through 3, and then p-values of 0.05, 0.01, and 0.001. So it's that big, which you can kind of see.
And the lab final is open notebook also.
Oh yeah, yeah. It's open notebook, like it's open notes. The real struggle with the lab final is honestly, bring food. Bring food, take breaks, go in the hallway. Like you can take the whole 3 hours. I'm here. I'm gonna be playing games on my phone or something. So like, it's no problem for me to hang out. Just take your time. But it's hard to work for 3 hours on something like this. It's very frustrating. You know that if you're doing your PhD, they have a thing called the qualifying exam, and you do 2 days. You know about this? It'll be like a 6-hour block of you're just taking exams, and then you do it day after day. It's horrible. It's great. See, it's wild. Like, who can maintain focus for 8 hours on anything? Not most people. Maybe engineers have to. Maybe that's the thing. I don't know. Okay. Oh yeah. No, if you imagine if you have accommodations, like that would be a 6-hour block of time. So you want 6 hours to do this thing? I can't maintain focus on anything longer than, I can do like 2 hours for a movie. But nowadays, every damn movie is 2.5 hours long. Am I right, people? Yes. A nice intermission in the middle would be great. Hey, media check-in. I saw that new Fantastic Four. That was pretty good. I liked it a lot. I was like, I'm actually having fun in a superhero movie. It was fun. Pretty cool.
Better than the last one?
Yeah. Yeah, but I watched that one on a plane, to be fair, so I didn't get the full experience.
I was really disappointed on the last one because
Yeah.
it just felt like it was build up, and then like 5 minutes of fighting.
Yeah, I love when he touches the dude and the guy explodes, though. It's very funny. Yeah. Sick. Fun. Okay, media roundup has concluded. Thank you all for listening to the movies that I've watched in the last week. Are we ready? I feel like we're ready. Any other questions about the lab final or whatever? Okay, this is the end, so we just gotta it's hard to not fizzle out. You kind of gotta ramp up for week 5. I'm gonna give you lots of extra support as much as I can. Reach out, communicate with me if you're running into any issues. I will work with you, and we will get you all through Bio 105. We're gonna do it. We're gonna get 100% passing the class again. That's the goal here. Without me just changing your grades to passing grades. Which I'm just not comfortable with, sorry. Okay. We can just start week 5. Where is it? Week 5, summer 24. Show me that PowerPoint. Here we go. It's a big PowerPoint, so it takes forever to load. This is a 195 slide PowerPoint. However, I'm currently only planning on testing you through the material on slide 150. So, that's not so bad. We have an extra day for it. There's a lot of really cool stuff here, though. This is probably the most engaging stuff right at the end of the class when we start to talk about inheritance and genetics. You can relate it to your own life, the things you see around you. It's the culmination of the Bio 105 is being able to make predictions about what the next generation of organisms are going to look like. I've talked to you a lot about how it's all about being able to make accurate predictions, and we've been building towards understanding, making accurate predictions about that. Just one last chance. Anybody got questions, specific questions about mitosis or meiosis or something like that before we start week 5? Yeah.
A question on meiosis, reading the literature, and I came across the word non-disjunction.
Yes, that's awesome. We are going to talk about non-disjunction in this PowerPoint. Let me just find the slide. Right near the end, because it leads to, so here's a non-disjunction event. It occurs in meiosis when the homologous chromosomes or the sister chromatids don't separate. I used to have this slide in the meiosis PowerPoint, but I felt like it confused things more. But it's going to be really important in determining how we get aneuploidy in organisms. So sometimes things will have an extra copy of a chromosome, or they'll be missing a copy of a chromosome. This is where Down syndrome comes from. It's trisomy 21. We'll talk about that. And then we'll talk about aneuploidy of sex chromosomes. We'll talk about the sex chromosomes and their importance, and how if you don't just have one X, if you don't just have 2 sex chromosomes, you could be a viable human being, an existing human being, because the sex chromosomes are regulated and operate in weird ways that are pretty interesting. Yeah, so aneuploidy, non-disjunction, we're going to talk about it. It's super important. Of course, yeah. Anything else? Okay, listen, I'm excited. I'm really excited to get into the week 5 PowerPoint. This is my favorite PowerPoint. When I was first creating this one, I was like, I kept trying to cut it down, but everything is important. Everything matters. And I was like, I just can't really delete anything. So we got to take a lot of stuff. Thankfully, we made some room by not talking about cell signaling. Sorry for the cell biologists out there. Okay. Ready? Here we go. Molecular basis of inheritance. So first, we're going to go into a little more depth with DNA. We've been really just barely scraping the surface with any discussion of DNA we've had so far. Okay? We're finally going to really talk about it. We're going to talk about the molecular basis of inheritance, which is the DNA molecule, right? That's the blueprint that you inherit that encodes all the proteins, encodes all the, encodes all the proteins that are going to regulate what your phenotype is, okay? DNA, boom, 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 learning objectives. We have to talk about DNA replication, which is going on during the S phase of interphase, right? Let's talk about the players there. Thankfully, we've already seen them briefly in our discussion of the PCR reaction. That's like saying ATM machine, right? The R is already for reaction, but in talking about PCR. Okay, cool. Remember this? The polynucleotide, right? Here's the polynucleotide. It's a big sequence of nucleotides. They're all joined together in a polymer via dehydration synthesis. Remember all this stuff, all right? You've got this repeating backbone, just like in a polypeptide. The backbone is a repeating unit where every nucleotide is the same in that it has a sugar and a phosphate group. So the only difference in each nucleotide is the nitrogenous base. In DNA, there are 4 possible nitrogenous bases. A, C, T, and G. What is it? Adenine, cytosine, thymine, and guanine. I think that's right. Did I nail it? Points for me. I'm taking 50 John points. Because why not? All right. Not really. I can't give myself those. They're too sacred. Okay. There's 4 possible nucleotides in DNA. A, C, T, G. You don't have to memorize the names of them. All right? It's fine to just be at that level. Now, in DNA, it's a double-stranded molecule. So there's a polynucleotide chain going here and a polynucleotide chain going here. And each is held together through interactions between the nitrogenous bases. So we call this base specificity, where A always binds with T and vice versa. C always binds with G and vice versa. The whole thing's held together via hydrogen bonds, all right, between the nitrogenous bases. Technically, this is all review, but that was like a month ago. So, 2 weeks, but it's basically a year in summer school. Okay? So do I have any questions so far? Please. With the hand? Oh, yeah. So you talk about handedness of molecules, which is a phrase I haven't really used before. But they talk about right-handedness and left-handedness of molecules. And it's actually handedness comes up a lot in chemistry. And it has to do if things are spinning right or spinning to the left. And we say this molecule is twisted and it has handedness. But I would assume that phrase is probably dead. They used it all the time when I was a student. But I don't use it, so, maybe, yeah. The idea is that it's twisted, okay? So, yeah, I was going to make a joke, but I'm not going to make the joke. It's too bad. It's just a bad joke. I'm just going to shut my mouth, all right? The molecule twists like this, okay? Solid? See, question?
DNA will always twist in that right-handed direction, correct?
Yeah. It's like a right-handed molecule. But then handedness also relates to how light gets rotated through molecules sometimes. So it's all complicated. Let's not ruin our lives thinking about hands. You think about your hands too much, life falls apart, you know what I mean? Don't look in a mirror and don't look at your hands. These are the 2 rules to get through the trip that is life. All right. Here, there's another important thing, the anti-parallel nature of the molecule, okay? It's got a 5' end and a 3' end on each strand. It's going to be really important when we talk about DNA synthesis. Remember, we talked about this during PCR. You can only add new nucleotides to the 3' end of a growing polynucleotide because of energetics. I'll show you a slide that explains it in more detail later. But that means that it's really important that we know which end is the 5' and which end is the 3'. They get their names from the carbon in the sugar that projects the functional group. In this case, it's the 5' carbon that has the phosphate group attached to it, and it's the 3' carbon that has the hydroxyl group attached to it. Don't worry about carbon numbering. Worry about that in your OChem course. Okay? If one strand has its 5' end here, then the complementary strand has its 3' end here. The strands are not identical to one another, okay? But if you separate the strands and read one as a template and add the complementary bases, you will regenerate a copy of the DNA molecule. How are we feeling? Okay, sick. So don't get confused. Okay, I was going to do the joke. Don't get it twisted. All right, there we go. Anti-parallel does not mean that the strands are identical running in opposite directions. It just means that the 5' and 3' ends are at opposite directions. Do you think it'd be fun and quirky if I got like 3' tattooed on my elbows, so when I do this demo in classes, I'm like, look, 5' and 3'? Would that be cool? Do you think people would think I'm cool? All right. Replication, DNA replication is super, well, it would be commitment. It would be a commitment. And I think it would probably really hurt because there's like bones there and stuff. But I could get the bones removed first, I guess. Origin of replication. DNA needs to replicate. Why does it need to replicate? Because the cell's going to divide, and each daughter cell needs to have the same complement of DNA as the parent cell, right? At least in mitosis. In meiosis, there's still the replication step, but then there's crossing over and all these things. I talked to you about it so long that I'm going to focus now on DNA replication. S phase of interphase, right? We cool? We're cool? All right. Solid. Here's an E. coli cell. Here's replication in an E. coli cell. Prokaryotes have a single circular chromosome that makes up their entire genome, right? We've talked about this a little bit so far. Whereas in the eukaryotes, the genome is broken up into multiple linear chromosomes. Remember that from way back when? All right. So in an E. coli cell, there's just a single origin of replication in a prokaryote. The parent strands, the strands that were already there, are separated, and complementary bases are added. All right? And this bubble that forms sort of just expands out and expands out until it completely separates the DNA, the chromosome, into 2 new chromosomes. And these new chromosomes are half parental DNA and half newly synthesized DNA. The newly synthesized DNA is indicated with a lighter blue color. The parental DNA, or the DNA that was originally there, is indicated with a darker blue color. How are we feeling?
What happens? Never mind, I got it figured out.
You good?
Yeah.
Sick.
It's strictly prokaryotes.
So here's the great thing. In eukaryotic cells, it happens as well, but there are multiple replication origins. All right? So there's just a bunch of these bubbles form. So it's the same sort of process, and the same types of enzymes are involved in the process. It's just in the eukaryotic genome, there are a bunch of linear chromosomes. All right? And because the chromosomes are long and linear, they need to be replicated differently. I'm just going to breeze past that pretty, drastically. So there's a single origin of replication in the prokaryotes. You get a single, what's called a replication bubble, all right, where the DNA is being separated and new complementary nucleotides are being added, and the replication bubble keeps expanding. The point at which the parental DNA is separated is called the replication fork. And the replication fork is where all the new DNA synthesis is happening. And we're going to talk about the enzymes acting at the replication fork quite a bit. Yes?
Will you restate that?
Yeah. Which part?
Um,
Hey, everybody. Week 5 of Bio 105. Sorry, being ridiculous.
Sorry, the point of something, and then you said it is the replication fork.
Oh yeah, absolutely. So the point where the parental DNA strands are being separated, that's the replication fork. And that's also where new DNA synthesis has to be happening. The DNA strands have to be separated so the complementary bases can be added. And we're going to talk about all the enzymes that are doing work at the replication fork. All right, today. Oh, go. Yeah, yes.
Sorry.
I'll pop right back to you, yeah.
Both eukaryotic and prokaryotic cells have a replication fork.
Both prokaryotic and eukaryotic cells have replication forks and replication bubbles, and the enzymes that are acting are very, very similar. Awesome, right, one ahead of you, yes.
If we're thinking about like human genome, would there be 23, what do we say, origins of replication? Would that be, is it as simple as starting at the beginning of 23 linear strands and going straight through? Or
Of course it's not that simple. It's never that simple. Each chromosome has multiple sort of origins of replication.
is that like splicing
Yeah.
together small pieces and putting them together as a whole process?
There is kind of. So check it out. Look at how this, Look at how we got the parental DNA strands, and they separated a bunch of different points, and new strands are generating, right? So you don't have to chop up the parental DNA, but this new DNA, it's got to get sewn together. And that's going to be a whole important aspect with DNA polymerase 5, I think, and DNA ligase. Yeah. I forget which DNA. There's DNA polymerase 3, DNA polymerase 1, DNA polymerase 5. They all matter, but some are involved in repair, some are involved in synthesis, some are involved in removing. It's a whole nightmare. Don't worry. We're going to talk about it. We're going to live through the nightmare together. All right? Please, yes, next.
When DNA is replicated, the replication fork is where the material comes from.
The replication fork is where the new material is assembled. The nucleotides are sort of like floating around. There's another enzyme responsible for charging up those nucleotides. Because if you remember, ATP is used in this process too. But this is so annoying. I'm sorry, this is so annoying. Look, right here, this is basically ATP, all right, if you put 2 more phosphates on it. So there are charged nucleotides floating around that are going to be able to provide the energy for the synthesis of the new strand. But to get more to your question, the replication fork is where the new strand is being assembled, right? And where the parental DNA is being separated. But the building supplies are sort of floating around. Does that get at your question? Okay. Solid. Awesome. Okay. There's a term going on, a new term here. Semi-conservative replication. All right? DNA replication is semi-conservative in that in the new DNA molecule, you conserve one strand of parental DNA. Okay? So the new DNA molecule is half old DNA and half new DNA every time DNA gets replicated. And that's called semi-conservative replication, which is a key phrase. You need to know what it means when I say DNA is anti-parallel and DNA replication is semi-conservative. Those are a couple of key phrases that you gotta understand. But don't worry, I got slides about them. You ready to talk replication fork? Okay, cool. If this slide looks familiar, okay, it's because you've seen it before. We looked at this slide when we talked about the polymerase chain reaction. Because remember, PCR, it sort of uses heat to get rid of the need for a bunch of these enzymes. But in the natural system, you cannot just heat up a cell to make the DNA separate during S phase. It has to be carefully unzipped and then maintained in that separated form. You have to take good care of the DNA. It's the most important piece of the cell, really. You know, that's why you keep it in a little bubble in the center of the cell and tightly regulate what enters and leaves with nuclear pore complexes. It's so important, you use 2 layers in the nuclear envelope, right? Okay. The enzymes that are at play, ready? Helicases. Helicases, do I have a cool, did I input the freaking, I didn't copy and paste the dang. Come on, stuff of life stuff. Dang it. Okay. Helicase. It's the zipper. It unzips the complementary, it unzips the parent DNA. All right? Exposing new bases. Not new bases, sorry. Exposing the bases that the complementary base can be added. So it starts like this. It separates, all right, with the helicase. It runs down like a zipper. Here's the helicase right there.
The definition on the slide's a little bit. It almost seems like it's.
Yeah, it kind of sounds like it is the like the main structure.
The helicase?
Wait, sorry,
Well, it's just that the definition on the slide almost makes it seem like it's undoing the twist rather than separating the whole thing.
It's frustrating because this is probably, the helicases are a class of enzymes. When the strand is twisted, that protects it too, right? So it does have to be untwisted. It also has to be separated. So the class of enzymes, the helicases, do both. I agree that this should have an additional little block of text. And I have nobody to blame but myself. All right, single-strand binding proteins. They prevent the DNA from snapping back together. You've got 2 strands that are perfectly complementary to one another. That's why they were held together. And there's hydrogen bonds ready to form, right? So the DNA needs to be held apart so that you can still read the exposed nucleotide. Single-strand binding proteins, I think they're sometimes also referred to as single-strand stabilizing proteins. There's names. People love giving these things new names. They attach here and prevent the thing from snapping back together. Cool? Topoisomerase, I explained this to you before about how when this DNA untwists, it's going to put strain on the DNA downstream. I always demo it with a cable. I used to use my headphones, but now I'm on wireless headphones, you know what I mean? Because I want more radiation blasting into my brain because I want to turn into Spider-Man, all right? So remember, you're separating the DNA down here. If you do that, it puts tension down here. You gotta relieve the tension. You gotta relieve the tension. That's what topoisomerase does. So gross. It tastes like the bench top. You know what I mean? Which tastes like poison. Don't put things that are in these rooms in your mouth. There's a lesson for you. All right. So topoisomerase. This is one of the things that I think the Stuff of Life does best, is explain DNA replication. It's a highlight of the book. So if you've got one of my loaner copies or you're checking out the PDF, zip to that part and I think you'll like what you see. Primase, next one. It adds an RNA primer, okay? Now, you remember primers from PCR. The primers indicate where DNA polymerase should start synthesizing the new DNA, right? The primers target a specific sequence. You need the primers because they provide the 3' end here. You need a starting little sequence of nucleotides that you can add on to. So the primer gives you a jumping off point to start synthesis of DNA. Question?
Would there be one on the other side going the other direction as well?
Points. Yes, you get points for sure. I think like 3 is reasonable. Just like the 3' hydroxyl group that's essential for synthesis of a new strand of DNA. There would be one here, and it would be going that way. Because DNA synthesis always has to go in the 5' to 3' direction. If this is the 3' end, this must be the 5' end. If this is the 5' end, well, this is the 3' end over here, then the 5' end must be here, and the 3' end must be here, and synthesis will be going in this way. Cool? Awesome. And that's going to be very important, because one of these is being synthesized in the same direction that the DNA is being separated in, and the other is being synthesized in this direction. So as new DNA gets separated, you're going to have to add another primer here, and then go boop, and then add another primer here, and then go boop, and then join all those chunks together, and get rid of the RNA, because the primer is RNA. And if, if you want to duplicate a DNA molecule, and you have RNA nucleotides in there, it's not a very good job, of replicating a DNA molecule. And I'm sorry to have to hit this again as well, but remember, RNA nucleotides are different from DNA nucleotides. They have a different sugar, and they use uracil instead of thymine as a base, nitrogenous base. So they are different nucleotides. Here's a nice little summary figure of the different enzymes you need to know about. Okay. Oh yeah, it was 3 and 1. I was wrong. DNA polymerase 5 is, I think, the one that's involved in DNA repair. Okay, check it out. Helicase unwinds parental double helix at replication forks. It means it's separating them. Okay. Single strand binding protein stabilizes the single strand. It's a protein that stabilizes single strand, binds it. All right. Topoisomerase relieves strain ahead of the replication forks. Remember, this is a bubble, so there's a fork on both sides of it. All right. Primase. Oops. Primase adds the RNA primer. Okay. Okazaki fragments, we're going to talk about them, don't worry. DNA polymerase 3 is the polymerase that actually attaches and builds the new chain, the new polynucleotide. All right? Builds the new complementary strand of DNA. DNA polymerase 1 is responsible for removing the primers. So you put the primers in, and then you got to get rid of them. All right? DNA ligase is the piece that I mentioned before with your question. It's going to join all the little fragments together. And we're going to talk about Okazaki fragments because you'll see that like two of these discuss Okazakis right here and right here. So it's a concept that's important. It has to do with the leading and lagging strand, which is one of those concepts like independent assortment where just historically, students have a hard time with them. And that is okay. So ask lots of questions about this concept. Ask lots of questions about anything. Okay. Anti-parallel structure. I've really tried to lay the groundwork so that when this slide comes up, you're just like, oh yeah, I know all this already. All right? DNA polymerase needs that 3' hydroxyl group to build the new DNA. It also needs a template strand that it can read to add the complementary bases. So you need a primer to get started, and you can only synthesize DNA in the 5' to 3' direction. Yes, please. This slide? It should be right after this. I only have one. I mean, I downloaded this file from the Canvas page. Did you get your PowerPoint from here in the module? maybe. Canvas, if you're looking at it through Canvas or in a different presentation app, maybe it's not showing that slide.
It looked the same when you zoomed out, like when you weren't in like slide.
Huh, you're right, look at that. Well, you look at that. Is this an animation? Does this, because it's in the slide view. It's an animation, that's what it is. Check it out. Whoa. Yeah, that's what's going on. Because when I hit next, it doesn't flip to a new slide. I put an animation that I added in here. I don't know why I did that. I thought it was cool when I built the PowerPoint initially last summer, because it has to do with anti-parallel nature stuff. Yeah, so this slide is there in the file. And also, hey, don't forget, folks, you all get free Microsoft Office through your tuition. And I really encourage you all to get it, put it on your laptops, do it. Because you're gonna hit a class where you're running your Excel in Google Sheets, and you come to me and you say, John, I can't get it to analyze the data. And I look at your output and I'm like, that's weird. Why isn't it working? And then you open it in Google Sheets and I get really angry at you. I'm a freaking jerk in my 300 level classes. I'm the worst. I totally change. I take the mask off, all right? I'm a bad person, okay? The point of the matter here is that, hey, why not use free PowerPoint? But I'm not trying to come down on you. It feels like I'm really giving you a hard time. Do what you want. Follow your bliss. Do your statistics in Google Sheets. Whatever. It's all fine. They're going to make us all start using Google for everything anyways. Microsoft will be bought by Google and it'll all be one product. I can't wait. Can't wait. I'm really happy. I love when they just delete my files. I love when they delete 2 years of lectures that I recorded and uploaded to YouTube because they said I had too much storage. Love it. Thanks, Google, if you're listening right now, and I know you are, because this video is going to go on to YouTube. Ah, what a world we live in. Man, I'm pissed. Okay. DNA polymerase. Ready? It's great. This is exciting stuff. This is great. Biology's, Google can't buy this. Anti-parallel structure of DNA elongation. You need a 3' end. You got to have the primer. How are we feeling? We cool with that? All right. Need a template strand. You can only synthesize DNA in the 5' to 3' direction. And then I've got this point right here that I've been bringing up since we first talked about DNA. Each strand of DNA is not identical to the other strand of DNA, but each strand predicts the structure of the other. You see what I mean? So one strand and the other are not identical, but they are anti-parallel, that the 5' end and the 3' end are opposite to one another. Okay? And then I put this abstract up to emphasize that the 2 strands are not identical to one another, because in this example right here, in this research, they're talking about 2 genes, 2 separate genes, one encoded on this strand of DNA and one encoded on this strand of DNA. So they're right opposite one another. All right? So that, pretty clearly demonstrates they are not copies. And why do I hit that so hard? Because people get confused about anti-parallel and what it means. And I don't want you to go out into the world confused about what the anti-parallel nature of DNA means. All right? Cool. You've seen this slide before too. All right? This is the explanation. This is the, explanation of how why DNA synthesis must go in the 5' to 3' direction. The nucleoside triphosphate is essentially a charged version of each nucleotide, okay? And it's like ATP, and GTP, and TTP, and CTP, and UTP, all right? It's basically an energy source itself, so it can drive its own polymerization. So you can't add to the 5' end. That's not how the enzyme works. But you can add to the 3' end. You break off these two phosphate groups. Two inorganic phosphate groups joined together are called a pyrophosphate. Listen, it's because light is emitted. A lot of energy is emitted when you break off a pyrophosphate. Heat gets emitted. Okay. There's an enzyme called luciferase, which is a wild name for an enzyme, where you can make this reaction produce light in addition to heat. And that's one of the ways that they used to do, or that they still do, DNA sequencing. It's fascinating. Is that anything I'm ever going to test you on? No. But it's fun to know that there's an enzyme called luciferase, right? What's that? Yeah, absolutely. And there's heat and there's light. Yeah. Anyway, if this doesn't totally click, that's fine. It's good enough to just know DNA synthesis has to go in the 5' to 3' direction. Yes?
I tried to ask a question a couple of days ago about like what powers DNA replication.
Yeah.
I made kind of an answer that I think it confused my mind about.
This is what I remember that question. This is what I was trying to tell you, was that they're like ATPs.
I think I got some. There's a part of my mind that's tempted to say like, so are the nucleotide bases the same structure as ATP, and they're like somehow in place of the ATP molecule itself?
I mean, the short answer to your question is all the energy is in those phosphate bonds anyways. So the additional structure doesn't actually matter. But let's see. We're just going to do ATP versus nucleoside triphosphate. Let's look at their different structures. Adenine.
I can tell specifically what's in my mind is like, does ATP like turn into a nucleotide and just attach itself to the end of the strand? I feel like I know that that's not accurate. Or is that accurate?
It's pretty accurate. It's pretty accurate. Yeah, I just want to see it. I would love a good image that compares the two. There should be a comparison. Yeah, okay, this is good enough. Right-click, open image in new tab. Right-click, open image in new tab. This is pretty good. What's that? It'll give me the, yeah, it's no good. You're right. Sometimes it gives me a nice big one. Oh, these are just crappy small images. They're all crappy small images. To begin with, 975 is our best one. No. It's okay. It's okay. It's okay. It's good to doubt people at the front of rooms who are speaking with authority, because sometimes they're wrong and very bad. All right. Sick. So the structure is basically identical. Here's a nucleotide structure, okay? And here's ATP's structure. It's got a ribose in it, right? Now, and adenine, nitrogenous base. Now, what's the difference here? Really, there's nothing beyond, in DNA, this is a deoxyribose. So this is missing an oxygen group. So it's a little different from ATP, but it's a little different from the nucleoside triphosphate with adenine on it, okay? The base is the same. The sugar is slightly different. The triphosphate chain is the same, all right? So it's basically ATP turning into a nucleotide, except that ATP is more like an RNA nucleotide than a DNA nucleotide, which, if you think about the idea that RNA is the first molecule to evolve, not DNA, makes a lot of sense, right?
So is that accurate that in this process, ATP is being consumed and added into DNA, like onto the chain of DNA?
It's not, because ATP has a ribose in it, not a deoxyribose.
So it's just providing energy. It's not like part of the structural component.
ATP isn't providing energy here. except for indirectly in charging these nucleoside triphosphates. These are providing the energy for DNA polymerase to act. It's coupling the cleavage of these phosphate groups to that exergonic reaction to the endergonic reaction of adding the new nucleotide, right? It's an anabolic and a catabolic reaction being coupled together. It's an exergonic and an endergonic reaction being coupled together. It's following all our rules of energetics because everything has to follow those rules of energetics, right? But this right here, this A base, was never ATP because ATP is missing a single oxygen atom on its sugar, right? So it's very frustrating. Yeah. But if this was a Biology 103 class, I would be like, yep. I was like, yep, that's it. This used to be ATP. Because, you know, taking Biology 103, it's never going to matter.
If these molecules are reassembled from basic forms of what used to be ATP, like if they have the same, I don't know, like little basic building blocks that are now just in different forms.
Well, everything does.
Well, yeah, of course.
You know. Yeah. Yeah, I mean, the bases need to be assembled. The bases are floating around. They're everywhere. The body builds up nitrogenous bases. The body needs nitrogen to do so. That's why you have to eat all this protein to get the nitrogen atoms out. Yeah. Was that a satisfactory answer for your question? Okay, cool. I got it. What is going on with the clicker? It's because I think it's because the PowerPoint is so big. This is good. Just not responding. Everybody, download PowerPoint and use PowerPoint because it's great. Gosh. Yeah, it's a big file. It's a big old file. It'll just be like, this file's too big. It'll make your computer crash. Yeah. So it's not that big.
Have you ever had it when you're like grading students' homework files and they turn in this massive file?
Yeah. Yep, it happens. That's why I don't use a computer that the school provides. I always just buy a gaming laptop at Costco.
Yeah, I
because
use my personal laptop
you got to do it.
for grading.
Yeah, it's essential.
But I don't want the massive files on my
Yep, you got to download them, then delete them. You know, it'd be nice if you could look at them all through Canvas. I could talk to you for hours about grading and downloading student submissions and all that stuff. But you know, I'd way rather talk about this. I've only taught it, 15, 16, 17 times. So, I'm very excited for discussing leading and lagging strands. This is a concept that everybody hates. And I am prepared for you to hate it as well. But, as I've done every summer and every fall and every spring, I will struggle through the concept with you and answer any questions you may have. Of course, that's literally my job. That's why they pay me all this freaking money. Yeah, it's great. This is a Bonobos shirt. No big deal. I could afford one of them. It's pretty cool. All right, check it out. Leading lagging strands. So DNA separation occurs here, right? It's going to continue unzipping in both of these directions, okay? That means that this strand of DNA is growing in the same direction that the DNA is being separated in. So it can be synthesized continuously. It's our leading strand. It's synthesized more quickly. You don't need to pop as many primers in. It's like, yeah, DNA polymerase, eventually you might want another primer. There's more complication to this, of course, right? But basically think about it like this. You put one primer, and this can just run all the way until it hits its next replication bubble. All right? Pretty nice. Same thing on the opposite strand going in this direction. The DNA is going to continue to be separated right here, all right? And so the synthesis can go in the same direction, all right? But this right here, okay, is the 3' end of the DNA molecule. Therefore, I attach a primer here, 5', 3', all right? I have to synthesize in this direction, away from the replication fork. So every time the replication fork opens up a little more, we have to slap a new primer in there, then synthesize in this direction, okay? All right? Yes, please.
Being opened up from left to right.
It's being opened up at each of these replication forks. Yeah, so this bubble is just expanding in both directions. Awesome. Yep. So each replication fork has all those enzymes at play, separating the DNA, stabilizing it, untwisting downstream, all that stuff. So you have to keep adding new primers and then running the DNA molecule up to that previous primer. And DNA polymerase can't take the 3' end of one and join it to an already existing exposed 5' end. All right? Plus, these are RNA, so they need to be yanked out. This is going to generate these little fragments of DNA that are actually not continuous. So, there are enzymes to replace that. Let's see if I've got anything here. I've got a video. Let's watch the video right now. Oh, let's watch the video. It's 4 minutes. Can I zoom in on it? It's going to run through leading lagging strands as well. And while I'd like to think that I do the best job of explaining it, this could be very helpful. Would you mind hitting the light for us again? So you get for sitting in that seat, I guess. All right, ready, folks? DNA replication. Let's crank the volume up a little bit. Is that about right? Do you like that noise? Sorry, I'm tired.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. Let's zoom in on one of them.
Whoa!
Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces. First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA.
You gotta
However,
do the whole thing, man.
connecting the pieces. An enzyme called DNA ligase joins the pieces together. Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
Nice, huh? You mind hitting that light for us? Thank you. Yeah. Cool. So there's the leading lagging strand. Oh my gosh. Come on, John, just hit the button once. There we go. Great. So there's the DNA replication video for you. Question, please.
You said on the top of the parent DNA, the 3' end was on that side?
Yeah, this would be the 3' end, and this is because this is the 5' end of the primer.
Okay.
Yeah. Yes. Cool. All right, well, it is definitely time for everybody's favorite, the 5-minute lunch break. All right, it's not lunch, but it's a break. Have your 5-minute break. I hope you enjoy it, and we'll be back, and then we'll just talk more DNA replication. Before you know it, we'll be able to talk genetics, and that'll be the exciting part, you know, inheritance patterns and all that. It's going to be really cool. Hey, recording world, we're back, and we're starting with a thrilling question. What do you got?
I felt like I was understanding the 5' and 3' end until that last slide about the leading and lagging strands. So
Okay.
if it can only elongate in the 5' to 3' direction, so if that top part is the 3' end, but then it requires 3' to elongate the DNA.
Yeah.
Can you explain that part more?
Yeah. So, yeah, I'm going to paraphrase because it's a really good question, and I want anybody who's... I'm going to paraphrase. It's a good question. I want anybody who's watching to hear it too, right? You're asking for clarification about the 5' and the 3' end and how a 3' end is needed to extend. How does that fit with the previous statements? Is that sort of accurate?
Yeah.
Okay, cool. So here we go. We've got... I'm going to actually not rely on the PowerPoint image, and we're just going to do some classic like Blackboard work. Sound fun? Okay. So here's the DNA strand. It's anti-parallel. Right? So if we separate it into two strands, and I know you said you were comfortable with the concept until we hit the leading lagging. That's really standard, but I'm going to make sure that everybody's comfortable in before we hit leading lagging with this piece, okay? So then we've got the 5', the 3', the 5', the 3', and the 5'. Okay, now this is your template DNA, and to make new DNA, you have to put an RNA primer in. Okay, that'll have its own 5' and 3' end. Okay, and then over here, let's put another RNA primer that has its own 5' and 3' end. So far, so good?
Yeah.
Question?
Is it true that primers don't attach to the very ends of the DNA strands?
It is true, because this is going like it's continuing, okay? So the DNA strand actually, and this is the piece that I'm going to add on in just a second, is the DNA strand's actually continuing down the end. So we're just going to look at this real quick. But they don't add on to like the end. You know what I mean? It's annoying. Yeah. The PCR quiz has issues. So here's the RNA primer, right? DNA polymerase is going to come and synthesize in the 5' to 3' direction.
So the 5' to 3' end of the primer, not the parent strand.
Not the parent strand.
Okay.
Does that kind of make sense?
That helps, yeah.
Solid. So this is the direction of DNA synthesis.
And then as far as requiring the 3' end to elongate, that also is referring to the primer.
The 3' end to elongate is also referring to the primer.
Okay.
When we're talking about DNA elongation and all that, we are talking about the new strand of DNA.
Okay.
All right, not the parent strand. Awesome, we got there. Okay, now I'm going to draw this out a little more, okay? Now, imagine, I'm going to delete the primes and add them on again. Now, we're looking at a replication bubble where the parental DNA comes back together. And now, these still have the 3' end here, the 5' end here, the 5' end here, the 3' end here, okay? So as these DNA strands separate, the leading strand can continue to be synthesized continuously, right? But you're going to have to attach a new primer here, a new RNA primer with a 3' end, and build new DNA like that as the replication fork continues in this direction. Does that sort of make sense? And this is continuing to be continuously produced. So this is the leading strand, the one that's produced continuously, and then this is the lagging strand, the one that's being reproduced in pieces. So at each fork, there's a leading and a lagging strand. It's really confusing, though, because this is the leading strand over here. See what I mean?
That makes a lot of sense.
This made sense?
That one statement saying that there is a leading and lagging at both ends, for me,
That helped? Okay, yeah. The leading and lagging strands exist at the replication forks. That's where the leading and the lagging strands matter. That is key. Awesome. I love finding like a thing that helps to bring the concept together. I got one ahead of you back there. Yeah.
So it just keeps like repeating itself.
This process, the lagging strand, yeah, it basically just keeps going like pop it in and then go, and then pop a new one in and then go.
Okay.
Yep. And then there are multiple bubbles, each with 2 forks, and they all join together, right? That's eukaryotic genome replication. It doesn't have to happen, and and prokaryotic as well. But yes, please.
and then for those leading and lagging strands, so the leading strand will always be in direction of the 5' end, and then so in theory, the one that's leading on one side will be lagging on the other.
Yes, if I'm understanding correctly, it's leading on this fork, so it's lagging on the other fork, because that strand always has the 3' end on this side and the 5' end way over here. Awesome. Okay, I think that worked. Okay, now let's lower the screen, reactivate the projector, and see if that resolved any misunderstandings that are still lingering about this image. Are there any further questions about this slide here? Yeah, 1 then 2, yes.
I think it would just help on that slide to have the 5' and 3' ends labeled on both
Definitely.
the parent DNA, but also the leading and lagging strands.
Absolutely. Yeah. No, I think I have a slide that does that in the spring version of this slide. But I have so many versions of this exact PowerPoint, things just get jumbled. So if I could write on this board, on this projector screen, I totally would. And I'd be like, okay, well, we've got the 3' here, the 5' here, the 3' here, and the 5' there. All right? So if you're annotating slides for your notes, that's a thing to annotate. And apologies, because usually there's a little red text that animates onto that slide at that point. Okay. We got to talk about telomeres. We have to talk about how every time a DNA molecule replicates, it shortens. Okay? Now, this is not the case with a circular chromosome, but with linear chromosomes, they shorten over time. And this is pretty confusing, but I'll try to get you through it. Because this is fundamental to understanding aging in eukaryotic cells. All right? Check it out. So you have your leading strand, your lagging strand. That bubble keeps extending, that fork keeps going, and eventually you hit the end of the strand. So you've got the 2 joined strands together, right? And you're separating them. And eventually you hit the end. And the 2 strands, are sitting there, right? The leading strand runs all the way up to the end of the template strand and just keeps adding new nucleotides, and eventually it goes, oh, we're done, and it pops off. But the lagging strand, you had to keep adding RNA primers all the way to the end, okay? You had to keep building these little pieces. So at the very end of this strand, this part of the parental strand, all right, you added an RNA primer, and then you remove that primer, but you can't replace them with nucleotides because there's no 3' end here, and you can't add to the 5' end here. So you take that primer out, and now there's just a little hanging piece. All right? So then when you replicate that strand, and you replicate this as the parent strand, well, it's shortened. And then every time you replicate, one of the strands gets shortened. All right? So you keep shortening and shortening and shortening the DNA every time the DNA gets replicated. Each chromosome gets shorter because at the very end of one of those parental strands, you had to throw a primer, and then you pulled the primer out, and the enzyme has no way of replacing those RNA nucleotides with DNA nucleotides because there's no nucleotide with a 3' end hydroxyl group available. All right? And the enzyme does not work to add nucleotides to the 5' end. It's not how the enzyme operates. Okay? Now, why does it work this way? Well, because it's the way it works. It's the way it evolved. Evolution is an opportunist, right? So if it works, it works. Now, there are organisms that get around this by just having a special enzyme that adds new nucleotides. So there are enzymes that can do this, all right? We have genes for them, and we express them in our germ cells so that gametes always have full-length chromosomes even if the germ cells are dividing, okay? But we don't express them in our other tissues. And one of the reasons is this prevents cells from dividing out of control. If a cell becomes cancerous and it's just dividing, well, sometimes the telomere shortening will just kill the cell off. It'll only divide so many times before it stops, and you're good to go. The problem is that in aggressive and bad cancers, usually they regain the ability to make telomerase, which is the enzyme that lengthens these endings. Okay? It's fascinating stuff. Don't worry, I've got that terminology coming up for you right here. So, you keep shortening the DNA every time. Eventually, the chromosome reaches a critical length and it won't divide anymore. Now, the chromosome, the organism controls for this by having the ends of each chromosome have a bunch of nothing nonsense DNA that doesn't encode anything. So if the DNA can get shorter, the chromosome keeps shortening, but it doesn't lose any important instructions. All right? It's like a book that has a bunch of blank pages at the front and at the end of it, okay? Because you loan it out a lot. But people are jerks and they, damage your book all the time. Every time you loan it out, they rip off a page from the front and the back. And eventually, you get to the point where they're ripping out essential information. And that will usually trigger apoptosis, and the cell will die. Okay? Or it'll go into G0, where it just doesn't divide anymore. And that's aging in a nutshell, yeah.
Are those blank pages the telomere?
Yes, those blank pages are the telomeres. Telomeres. I don't know what the correct pronunciation is because in science, people just pronounce things and it's infuriating. I've heard both. Cool. I think like television, telomere. Telovision. Telovision? You ever watch Telovision? When I was a kid, we weren't allowed it. No, we had a TV. It was great. It was great. No cable. We had a TV. Why am I telling you this? Because I'm tired. I'm so tired. Are you all so tired today? Yeah. This is rough. We got 30 minutes left. We can get through this. Okay. Telomeres. Telomeres are the non-encoding ends, okay, of the chromosomes. There's a bunch of repeats of T, T, A, G, G. Doesn't encode anything. It just sits there and gets shorter and shorter every time they divide. Okay? Postpones the erosion of genes that are located near the ends of DNA molecules. So the shortening of DNA is not prevented, but it's postponed by having those blank pages at the end of the book. Okay? Telomerase is an enzyme, and what it essentially does is it lengthens, we're not going to worry too much about this, okay? It synthesizes a new complementary strand of, RNA, and then it lengthens the 3' end of one strand so that the other strand can be lengthened. Don't worry about the details here. It's an enzyme that rebuilds telomeres. It's called telomerase, or telomerase, either way. Plenty of organisms have it and express it and don't age. All right? It works for them. Are rates of cancer higher in those organisms? Yes. Have there been experiments with mammals to get them to express telomerase in their tissues in order to reverse aging? Yes. Have those organisms developed pretty aggressive cancers in a lot of cases? Yes. All right? These are all great questions, okay? The second one of you reverses aging or stops aging, call me up, all right? I'd love that. I want to live forever. I don't care, I want to do it. I'm into it. Give me that pill. Give me the pill, all right? After you've tested it on somebody else, but, keep me in mind, just in case it's one of you. I'm hedging my bets. It's the whole reason I got into this field. Someone will keep me alive forever, right? All right, cool. Let's keep on moving forward. Telomeres, questions about them? Sick. I love it. Elizabeth Blackburn, Nobel laureate, discovered how telomerase works. Pretty cool. Thank you. Now figure out how to make me be 28 again, because that was a sweet time. 30s are best. No offense. 30s have been the best. Okay, good questions. I laid these points out earlier. All right. There are also repair mechanisms at work here. There's proofreading stuff. Okay, here's some proofreading DNA, or this enzyme is proofreading DNA. All right. So sometimes you accidentally add the wrong nucleotide. DNA polymerase screws up and adds the wrong nucleotide. Oftentimes, the system can catch that and replace it with the appropriate nucleotide. There's another DNA polymerase involved. It has a number after it. DNA ligase is involved. One of the ligases. It's a whole thing. It's a bunch of enzymes. All right? So one way that the DNA proofreading or the DNA repair machinery can work is by detecting bumps in the DNA that occur because of mismatches here, like T to C here. Those nucleotides won't bind at all, so they repel each other and they make a little bump there. All right, and the machinery can just detect the width of the DNA double helix, essentially the DNA, the double-stranded molecule. Okay. Also, this example here shows DNA repair of specifically something called a thymine dimer, which happens when you jazz up the DNA too much and the strand sort of goes like, and the hydrogen bonds get weakened and it goes boop, like that. And two of the adjacent nucleotides get too friendly with each other. It makes a little bubble. It wants to lay flat, but it doesn't. So those are called thymine dimers. You won't be tested on them, but they happen a lot after ultraviolet damage. Because ultraviolet radiation is high energy radiation. It can lead to problems. Then when the DNA gets replicated, you get the wrong nucleotide added. It's a fascinating thing that we're not really going to worry too much about. That's all the DNA replication stuff that we needed to talk about. Cool? All right. Transcription, translation. We're finally getting into the central dogma. All right, I've been drawing it on the board since the first week of class. And remember back then, none of us knew what to expect. I was like, who's gonna be the jerk in the class who makes my life hard? Turns out it was nobody, and you've all been just a dream to work with. You were all like, oh, I don't know about this guy. Well, that's what you're gonna do. And you haven't even noticed how many of your classmates have disappeared, you know? But I'm happy that I seem to have turned out great for those of you who, have stayed off the list. I'm not killing your classmates. I'm sorry, I shouldn't joke about that. Sorry. All right. Simpler times though, right? When we first started talking about the central dogma. Now it's coming back, baby. We're talking transcription. We're talking translation. You're gonna love it. First, transcription. What is it? DNA is read, and it is used to direct the synthesis of an RNA molecule. Transcription. Okay? Translation, the RNA gets read by a ribosome and is used to direct the synthesis of a polypeptide. Just like how everything's simpler in prokaryotes, the central dogma is simpler in prokaryotes. Okay? They're simpler. Don't pity them. They're everywhere. They're doing just fine. Okay? In a eukaryote, the process is more complicated, but that helps to add to the process is more complex, which helps to aid in us developing all this complexity, which lets us build my phone, which I love. I'm very into it. Okay, bacterial cell, check this out. There's no different chamber for the DNA. That means that transcription and translation are both happening within the cytoplasm, and they can actually co-occur almost. So while transcription's happening and mRNA is being produced, translation can start, all right? And that mRNA can be read by the ribosome before it's even done being made, okay? However, in the eukaryotic cell, transcription occurs within the nucleus, all right? So the RNA needs to be fully produced, and then it's actually going to be processed still within the nucleus, okay? And then after processing, it's going to be exported into the cytoplasm or into the endoplasmic reticulum. Let's not get too confused here. Remember how the endoplasmic reticulum has ribosomes on it? So you know that some of the mRNA is going to those ribosomes. Let's not panic too much. Just think about it this way. It has to get exported from the nucleus where it can hits a ribosome. Okay? The ribosome reads it and makes the polypeptide. Cool? Yes?
I should have this from basic Chem 105 or whatever.
That was forever ago.
It's synonymous with protein or like building blocks of protein?
Polypeptide is synonymous with protein. Yep, a polypeptide is just a polymer of amino acids. It's called a polypeptide because it's a peptide bond that joins them. And it's a peptide bond because that's what we call it. And it's a bond between amino acids. It's all cyclical, you know. Where somebody defines one thing and then all the other words build up around it. You know what I mean? It's funny. Yeah.
What happens during the RNA processing?
Absolutely. There are two important processes that happen. One is called RNA splicing. I wonder if I just have the slide. The slide's coming up in a little bit. One is splicing, where some pieces of DNA some pieces of RNA are removed. Okay. And then the other pieces of RNA get joined together. So you remove these things called introns, which are intervening sequences, and you splice together these things called exons, which are the expressed sequences that actually encode a protein.
So does that not happen in a prokaryote?
It does not happen in a prokaryote. Yep, that's awesome. 3 points. That's really good. This RNA processing step, you'll notice it is not in the prokaryote. This is a eukaryotic thing. It gives us more flexibility where like a single gene, a single piece of pre-mRNA can be mixed up in all sorts of different ways to make different full mature mRNA. All right, but don't worry, I've got whole slides about RNA processing. The second thing that happens during RNA processing is modifications to the end of the molecule, which in our Bio 105 world, what those do is they facilitate export from the nucleus, they add stability to the mRNA molecule, and they facilitate attachment of the ribosome.
Is that what the little yellow things are?
That's what the little yellow things are. It's a 5' cap and a 3' tail, you know? And don't worry, we'll talk about it. Yeah. Okay, here's central dogma again, very basic level. DNA, here's the template strand, 3' ends there, 5' ends there. Now, RNA synthesis, RNA is really similar to DNA. It still needs to be synthesized in the 5' to 3' direction, okay? It's still gonna work the same way, all right? That's the energetics of polynucleotide synthesis. If here's a 3' end, then the mRNA is going to have a 5' end adjacent to it. A to U, C to G, A to U, C to G, G to C, A to U, G to C, T to A to T, C to G, U to U, me to me. It's horrible. It's horrible. So here's transcription. You read a template strand of DNA. You produce a strand of mRNA. What's the M stand for? Messenger. Very good. There's other types of RNA that also get produced via transcription, but that don't go through translation. Things like tRNA, or microRNAs, or siRNAs, small interfering RNAs. There's all sorts of different RNAs that get produced by transcription. But only mRNA is used to direct protein synthesis and will go through translation. Okay? So we focus a lot on mRNA, and we don't really focus on the other types of RNA, though you'll focus on them if you go into genetics, continue with this topic, you know. Yeah, it's all very interesting stuff. We'll talk about tRNAs. That's the only other class that we'll really talk about. Okay. Do we need to talk about codons immediately? We do. We're going to talk about codons immediately. That's right. I rapid fire this stuff. I forget. Okay. So how do we feel about RNA synthesis in transcription? You see how you read the template DNA strand, you add the complementary base pairs, U instead of T, 5' 3' in, it still needs to be anti-parallel. Cool so far? All right. This is all carried out by one like workhorse enzyme called RNA polymerase. Brilliant name, right? Which basically does everything that occurs at the replication fork by itself. Okay? It's a big old mondo mega enzyme. Okay. The next step in the central dogma is translation, where the sequence of mRNA is read and is used to direct amino acid sequence. So this is how DNA encodes a protein. DNA encodes mRNA, which is then read by the ribosome to generate a sequence of amino acids, and that makes the protein. Okay? Now, the RNA is read in 3 base units. Okay? So there's a code going on here. Those 3 bases are called a codon. All right? So UGG is read by the ribosome and encodes tryptophan. All right? UUU is read by the ribosome and encodes phenylalanine. All right? So the mRNA nucleotides are read in units of 3. Now, there are 4 potential nucleotides, A, U, G, and C, okay, read in sequences of 3. And if you remember our independent assortment math, that means that there are 4 to the 3rd possible combinations for a codon. So there are 64 different possible codons that can be generated, okay? There are 20-something amino acids, okay? So how do 4 different bases encode 20 different amino acids? Well, it's by being read in units of 3, okay? So it's a 3-base code, a 3-unit code with 4 potential numbers in each unit slot, all right? Cool. How are the instructions for assembling amino acids? There's 20. I freaking always want to say there's 23. But that's the chromosomes. It's numbers. They all get mixed up less. Yeah. There are 20 amino acids. All right? There are 20 of them. There are 4 nucleotide bases. How many nucleotides correspond to an amino acid? It's 3. Okay? So there are 64 possible combinations. Here in this table is every single possible codon that could be produced in an mRNA molecule. All right? But the genetic code is what we call redundant, in that more than one codon can encode a particular amino acid. But the same codon, one single codon, only specifies one amino acid. So each codon is specific to an amino acid, but there can be multiple codons, different codons, specific for the same amino acid. So there are multiple codons that encode tryptophan, all right? Because there's 64 possible ones. Every single one is used, okay? And only 20 different amino acids. Reading frame, this concept's going to be super important. Codons have to be read in the correct reading frame, but I have slides to talk about that. But imagine that this G went away. It was just gone. due to some issue with DNA replication or transcription, well, then the first codon would be UGU. And the second codon, instead of being UUU, would be UUG. So losing a nucleotide here shifts all the codons and leads to drastic changes in the polypeptide sequence. These are called reading frame mutations, and they're incredibly dangerous and harmful to the organism. Don't worry, I have slides coming up all about it. Question? Or you want to wait for the slides? What's that? A codon is a unit of 3 RNA nucleotides. Yep. Here is a codon table. The codon table is used to convert an RNA sequence into an amino acid sequence. Now, you can cheat a codon table and just be like, hey, the first one is CAG. And just be like, I'm just going to look around here until I find CAG. There it is. All right, no problem. I forget which, it's not glycine. It's something like that, though. I forget what amino acid that is. There's 20 of them. I don't have them all memorized. Do I have a picture of them? Nope. Okay. Or you can use the table as designed to look them up. Let's say you've got a sequence. Let's just do this. I have to do this. Every time I try to be like, I don't have to write anything down right now. It's week 4. I'm tired. DNA sequence. Let's start with the RNA sequence. Make our lives easy. Okay? Here's the 5' end. Here's the 3' end. Let's go with U, U, G, G, C, A, C, G, U, A. Whatever. All right? If we start reading here, we read the codon like so. UUG, how do we look that up? Well, the first base is U. The second base is U. The third base is G. So see how it's U, U, G. Leucine. Leucine? Lysine? Leucine? I love leucine. GCA. GCA. Right there. Alanine. That one's easy. All right. Cool? You see how that works? Pretty mellow. Now you do read RNA. The ribosome reads RNA in the 5' to 3' direction, which is pretty frustrating because you got a bunch of stuff to memorize here. But it's like DNA synthesis goes in the 5' to 3' direction. RNA synthesis goes in the 5' to 3' direction. The ribosome reads RNA starting at the 5' end. So it's read in the 5' to 3' direction. It's 5 to 3 all the way down, but it can still get confusing. I'll show you a trick for remembering that, but it requires you remembering that both processes can occur together and at the same time in a prokaryote. Anyway, we'll talk about it. Okay. The coding sequence of green fluorescent protein, something that you're going to have to, do next week in our lab is interpret plasmid maps. And for the lab assignment, you'll have to, answer some questions about plasmid maps. But on the lab final, I removed the plasmid map questions because, I don't know. I think the plasmid map stuff is not especially interesting. And I already had 24 questions on the lab final. I was like, that's enough. Anyways, how long is the coding sequence of green fluorescent protein? Well, I've shown you plasmid maps before. The green fluorescent protein gene runs from base 1342 to 2062. And I've done this enough that I can just tell you that's 720 base pairs. I don't have to, do the math anymore, which is wild. And I never thought that would happen. So the green fluorescent protein gene is 720 nucleotides long. How long's the protein? You got it. 720 nucleotides per gene. All right. And there are, oh my God, for every 3 nucleotides, there is 1 amino acid. All right. That means that there are 240 amino acids per the gene. Okay? Or if instead of gene, we put protein, or GFP. If each amino acid weighs 110 daltons, per protein, how much does the protein weigh? Well, 110 daltons per amino acid. Amino acids cancel out. Gives you an answer in daltons per protein. I don't know, what's the number here? Times 100 will be 2400. 2640? 26,400? Something like that? Yeah, did I get it? Sick. Nice. No big deal. I can do mental math. But look, what's that? Dimensional analysis. Conversion factors. Everything is. Most math is. All right, at least in biology, chemistry. Okay, that makes sense? Sick. The reason that I always have this slide and always do that math with you is, 1, to reinforce how useful the dimensional analysis and the conversion factors are. I always like to reinforce that. And 2, because answering this question requires that you understand what the codons are. All right? Cool? Now, we're going to talk about translation, where the protein gets produced, and translation initiation and all that stuff. But you might have noticed that, hey, there's this AUG sequence that says, it's highlighted and it says start on it. So this little sequence right here is where the ribosome initially binds, and it's going to indicate like, oh, this is where a gene starts. Does that make sense? And then we have 3 options over here that are stop codons, and they tell the ribosome, stop, the protein's done. All right? So in addition to encoding a bunch of different amino acids, the mRNA also encodes like, this is where to start, and this is where to stop producing the protein. Does that make sense? The start codon's always AUG. Stop codons vary. This is the same in all living things. Okay? So the same nucleotides encode the same amino acids. The codons are the same. That's why you can take a bacterial gene and put it into a plant and get it to make a protein. Or a human gene and put it into a bacterium and get it to make a human protein. All right? So codon table's the codon table. You know? DNA, RNA, the central dogma, it evolved so long ago, and it's so highly conserved that it makes genetic engineering, like possible. And it also makes it very, very hard to evolve away from. Okay? It's just so ingrained. All right. Molecular components of transcription. We'll run all the way. We're just gonna run through the full last 5 minutes of lecture. It's 5 more minutes. And I really want to give us lots of time to talk about the fun stuff. Okay. Transcription. There's a big workhorse enzyme called RNA polymerase. It separates a very small part of DNA. All right, it separates the parental strands, reads the template, synthesizes in the 5' to 3' direction. Okay? Because it only has to, add a complementary nucleotides to one of the strands, not both, it means that it doesn't need as much stabilization. It can all be accomplished by one mega enzyme complex thing. That, of course, has a bunch of quaternary structure, so it's a bunch of polypeptides joined together, just like any complicated protein. RNA synthesis follows the same rules as DNA synthesis, same base pairing rules, okay? Also must be synthesized in the 5' to 3' direction, but uracil substitutes for thymine, so we get U's where there would be T's. See? Questions? Pretty mellow, honestly. Transcription's pretty mellow. Okay? Now there's more to it. All right? If you're interested in, like, you know, this is just a cool thing where the stuff of life talks about how an enzyme complex locates a certain gene, it gets activated, okay, and how RNA polymerase, here it is, zipping along and reading and making the copies, you know. It's kind of cool. Replication bubble, the transcription bubble here, so the dereplication bubble, and eventually RNA polymerase gets a terminator sequence of bases that tells it to stop working. This is different from the stop codon, okay? This is the terminator sequence. It's different. I'm not going to test you on terminator sequences, though, because I think that it muddies the waters too much. Okay, but of course there's more to it. This right here, these 4 little blocks explain the 3 stages of transcription. The 3 stages of transcription being initiation. It starts, RNA polymerase binds, starts making RNA. It elongates. The RNA transcript gets longer. It terminates. It hits the terminator sequence, and RNA polymerase stops making RNA. All right, so starts, makes the sequence longer, and then stops. Nothing too wild there, okay? So there's this promoter region. That's where RNA polymerase can bind, okay? It's what we call upstream of the transcribed unit. I think that is self-explanatory. Elongation, finalized right there. Now, RNA polymerase binding to the promoter is facilitated. This is a way that regulation can happen. If you allow RNA polymerase to bind, then you get the RNA. If you don't allow it to bind, you don't get the RNA. If you get the RNA, you can get the protein. If you don't get the RNA, you don't get the protein, right? So this promoter and binding of RNA polymerase, that's all tightly regulated by things that are, the first way that there's regulated that we'll have to know about are transcription factors. Basically, a transcription factor binds to a promoter and it allows RNA polymerase to bind. So it's a factor that encourages transcription. But we will start here on Monday with the stages of transcription. There's some transcription factors. We'll run through initiation, elongation, termination pretty quickly. We'll talk about mRNA processing. Then we'll talk about translation very briefly. We'll get into regulation of gene expression. Then we get into some cool mutations, Mendel, mathematics, Punnett squares, all this stuff. It's going to be a hoot and a holler. All right? Talk blood types, probability math, chromosomes, polyploidy, aneuploidy, natural selection. And then we'll be done. And then if we have extra time, I'll talk to you about some more cool stuff, you know, that we've got that's still interesting. Anyways, and then the PowerPoint ends. It's a lot of stuff. Great job today, everybody. Great job getting through week 4. We are technically on slide 27. And we have to get through like 100 and some odd slides next week. It's going to be fine. We've done it before. We've done more than that. Okay, come talk to me if you have questions. I hope you all have a splendid weekend. All right, get some recovery done. And I'm ending the recording there.